Okay, so it's now 11, so welcome everyone to the ERGA BioGenome Analysis and Application seminar. So it is my great pleasure to welcome Martin Mascher and Marina Marone. So Martin leads the Domestication Genomics Research Group at the Leibniz Institute of Plant Genetics and Crop Plant Research, where Marina is a postdoc. And so Martin is interested in studying crop evolution and adaptation and how these processes shape the genetic diversity of the domesticated plants and their wild ancestors. So today, Martin will tell us about structural variation in the pangenome of wild and domesticated barley. This will be the first talk of about 20 minutes. And then Marina will tell us about how to assemble plant genomes with Tritex. And so the floor is yours. Thank you again, both of you, for giving us the opportunity to hear about your research. And the floor is yours. Thank you.
Thanks, Jose, for the introduction. I will share my screen. Can you see my screen? Is it in full screen?
Yes, perfectly.
Okay.
Thank you.
Good. Yeah. Today I will tell you about structure variation in the pangenome of wild and domesticated barley. And yeah, as Jose said, I'm working at the Leibniz Institute of Plant Genetics and Crop Plant Research, IPK, in Gattersleben. And I first want to tell you a bit about our institute and our gene bank infrastructure, because this is central to our research aims and also was the basis for the research reported in this talk. So IPK Gattersleben is a publicly funded research institute. It's located in the middle of Germany. And yeah, our key infrastructure is the gene bank. That's the National Gene Bank of Germany. And the gene bank is basically a repository of seeds for plants, for cultivated plants. And our gene bank hosts 300,000 accessions. That means seed lots of 1,500 plant species. And gene banks can be considered living libraries. They maintain genetic diversity of crop plants and their wild relatives. So over the 20th century, researchers collected plant seeds from around the world, and the seeds are maintained in gene banks. And for most seeds, they can be stored in cold temperatures, and then they last for a long time and maintain viability. And our gene bank team regularly performs germination tests, and if germination rates are too low, the seeds are regenerated. That means they are grown in the field and harvested. So this is a way to maintain genetic material from a known origin in perpetuity. It is a large logistical effort, but the rationale for having gene banks is that they are living libraries of crop diversity and an important resource for crop improvement. But like real libraries that have books, the real value comes not from just holding the books, but also reading the books. And this is the great research challenge: how to activate the genetic diversity that is maintained in gene banks for crop improvement. And we adopted a genomics approach. Central to this aim is the genomic characterization of our gene bank collection. And with genomic characterization, we mean genotyping with high-density genome-wide markers at the level of entire collections, and then obtaining genome sequences for representative individuals of our collection. And using this genome-based characterization, we hope to understand patterns of genetic diversity and also identify beneficial variation that can be used in crop improvement. And in this talk, I will tell you about our efforts in barley. This is our focus crop at IPK. This is where we first followed this gene bank genomics approach. So barley is a cereal crop that is nowadays mainly used for brewing alcoholic beverages and also for feeding animals. But barley has a long history. It is one of the first domesticated crops. Domestication started about 12,000 years ago in the Middle East. And at that time, it was also used for human consumption, and it still maintains an important food crop for direct human consumption in some regions of the world, like the highlands of Ethiopia and Tibet. And I like to show this clay plate. This is one of the earliest remnants of human writing, and it shows a pictogram for barley. So already in ancient Mesopotamia, barley was an important crop and used for brewing, as we still use it today. And so in Barley, this talk is about gene bank genomics and pangenomics, and I want to show a workflow how we integrate these two approaches. So we start with our entire gene bank collection, and using genome-wide marker data, we make an informed selection of representative and diverse germplasm for genotyping. This can come from the domesticated barley, but also from wild relatives and more distantly related species. We then construct genome assemblies to obtain a catalog of genetic variation. So a pangenome that tells us about single nucleotide polymorphisms, insertions and deletions, but also larger structural variants like chromosomal inversions, duplications, and rearrangements between chromosomes. Still, as in other species, genome sequencing is expensive. The barley genome is 5 gigabases in size, so larger even than the human genome. And if we want to collect genomic sequence data for a large panel, thinking of hundreds or thousands of accessions, we cannot construct full genome assemblies, but we rely on short-read resequencing on the Illumina platform. And the ultimate goal of this data collection is to link genetic and phenotypic variation. So usually for the same accessions that we sequence, we also collect phenotypic data and are then able to use approaches like genome-wide association scans to connect genotype and phenotype, or using population genomics approaches, we can identify targets of selection. And then this pangenome and associated resources are a permanent resource for other researchers that can then also be complemented with transcriptomic and epigenomic data and be made accessible in a browser. So this is the outline of our Barley gene bank genomics and pangenomics approach. So let's go through the concrete results. So about more than 5 years ago, we published the gene bank genomics study in Barley. This is based on genotyping all 22,000 Barley accessions in our collection. And this resulted in a matrix of 50,000 markers that for each individual, or for one representative individual of each accession. And this variation matrix is represented here in the principal component analysis that can be thought of as a representation of the genetic diversity space of Barley. And the predominant pattern is something that has also been observed in, for example, human genetics, that genes mirror geography. The first principal components nicely correspond to geographic regions. So, for example, in the left part of the plot, we see barleys that come from Europe, the Mediterranean region, so the Western Eurasia, while the eastern plot or the right-hand plot shows barleys from East and Central Asia. And on the second principal component, barleys from Ethiopia stand out. And this corresponds nicely to the 3 directions in which barley, which originated from the Fertile Crescent region, the Middle East, spread out across Eurasia. So we use this representation of the genetic diversity space to make an informed selection of diverse and representative germplasm for genome sequence assembly. In the second phase of the Barley pangenome, we included 76 diverse genotypes, and I will describe how we selected them. So first, we selected 53 genotypes of domesticated barley, and we used analysis like principal component analysis and structure-like algorithms to find representative of all major germplasm groups of domesticated barley. And the selected accessions are represented here in this PCA. And you might wonder why we see here a cluster of orange dots. And these are actually barleys from the Middle East. And if we look at higher principal components, they occupy a larger diversity space. So they come all from the Middle East. I apologize for the colors. So the green region corresponds to the orange dots. And this is the center of origin and also the center of present-day diversity of barley. So traditional land races from from the Middle East have high genetic diversity, and we expect to find lots of new variation there that is currently not exploited in barley breeding. We also selected 23 genotypes of barley's wild progenitor. This is a species, or it's actually a subspecies of barley called Hordeum spontaneum, so spontaneous wild barley. And this is a grass that is very common in the Middle East. You can find it easily if you travel in this region. It's a common grass, and also our gene bank and other gene banks hold collection of wild barley, and using a similar approach, using marker data and population structure analysis, we identified a diverse set of 23 barleys that is also represented throughout the geographic range of wild barley. Then we used long-read sequencing on the PacBio platform, so hi-fi reads and chromosome conformation capture sequencing, Hi-C, to assemble 76 chromosome-scale genomes. And Marina will tell you more about our pipeline for genome sequence assembly in her presentation right after mine. And then we have these 76 genome assemblies. And in the next slides, I will show you some results from the comparison of these genomes at the genome-wide level and also at loci of economic importance. We at first aligned these genomes to discover structure variants, and they are shown in this gene space plot, which shows the collinearity between genes. And we can see that there are, for example, large inversions between the variety Morex and the landrace from our accession. For the first time, we also observed translocations between chromosomes in barley, because they are thought to be rare, because barley is a diploid, and translocations are much less well tolerated than in, for example, wheat, which is a hexaploid, so a polyploid species, where such translocations are rather common. We then used a graph-based approach, the single copy pangenome, to estimate pangenome complexity. This is not a full graph-based pangenome, but a simplified representation where we focus only at the part of the genome that is single copy in each of the genomes, and we then perform a clustering to get a quantitative estimate of pangenome complexity. And not unexpectedly, we saw that the pangenome complexity in wild barley is higher than in domesticated barley. So with only 23 accessions of wild barley, we cover a larger sequence space than with 53 domesticated accessions. And this is consistent with our expectation of higher genetic diversity in the wild progenitor and lower genetic diversity in the crop, which has undergone a domestication bottleneck. The next example I want to show you is for structure variation at a specific locus of agronomic importance. And this locus is the alpha-amylase locus. This is a locus of enzymes that degrade starch. And this is important in the brewing process. So when barley is brewed, it is malted, so the seeds are germinated. And during this stage, the alpha-amylase enzymes are released, and they digest the starch that's stored in the endosperm of the grain, and this releases sugar. And this sugar can then be used to, or is then used by yeast to ferment it into alcohol. So this is the central aspect of the production of alcoholic beverages. And accordingly, these alpha-amylase enzymes have been studied for a long time. So already in the 1980s, with the first PCR-based approaches, it was observed that alpha-amylases occur in high copy numbers, and there are several loci in the genome. And one of these is the ME11 locus. But all previous barley assemblies could not capture this structure variation and this copy number increase, because these copies are highly similar and were collapsed in previous short-read assemblies. So our long-read hi-fi-based assemblies provide the first glimpse into structure variation at this locus. And we were quite surprised by the level of structure variation we saw. So there's first copy number variation. So Morex is kind of a reference cultivar that we use. It has 6 copies, one of which is inactivated by transposon insertions. And then we saw copy numbers at this locus ranging from between 2 copies in a wild Barley FT11 to 8 copies in the Barley FT880. And these colored boxes with the numbers stand for different sequences at the nucleotide level. So in addition, or on top of structural variation, we also have sequence variation of the protein at the protein level. And our collaborators at Carlsberg Research Laboratory also did some AlphaFold modeling and found that these protein variants are likely to affect the functional domains of the amylase locus. But an important question is how does this genomic variation translate into breeding applications? Does this variation at the alpha-amylase locus really have an impact on the malting process? And to test this, our collaborators at Carlsberg Laboratory constructed defined genetic material in which the alpha-amylase allele was replaced. And so these are near isogenic lines in the background of the very successful malting variety RGT Planet, where the genomic region that contains the alpha-amylase AMY11 locus was replaced with different alleles from other barleys named Mark and Morex. And then they performed multi-environment, multi-year field trials, and they were able to observe that this allele replacement at AMY11 really resulted in different alpha-amylase activity. So this first experiment showed us that most likely this structure variation at this amylase locus can be translated into meaningful applications. And of course, the next step would be to test how this allele from wild barley, but that has 8 copies, performs in brewing, or maybe also how a barley that has only 2 copies performs in brewing. But so this is, of course, work for future research because the genetic material has to be developed, and then the field trials have to be performed in an agronomically relevant environment. Yeah, so this I just want to give a graphical summary of my presentation. We started from genetic diversity in gene banks of barley, elite material, landraces, and wild relatives, and constructed a representative quartet for further study. And then we performed genome sequence assembly and also aligned other data sets to get a catalog of structure variation in the pangenome of barley. And yeah, this is now an important resource for future crop improvement. So our research was published last year, and I want to thank my collaborators in the Barley Pangenome Consortium, which was coordinated by Nils Stein. And the lead author of the study was Murukati Jayakody, at that time a postdoc in my group, now an independent researcher at Texas A&M University in Dallas. And yeah, the work on the alpha-amylase locus was led by Christoph Doctor at Carlsberg Research Laboratory together with his postdoc Chong-Chan Lu. And with this, I'm at the end of my presentation, and I thank you for your attention, and I'm happy to take questions later after Marina's talk. Thank you.
Thank you, Martin. Wonderful. So, Marina, the floor is yours now.
All right, thank you. Can you see my screen? Is everything fine?
Yes, everything's fine.
Okay, 1 second. All right, so now I will talk about how to assemble plant genomes with Tritex. That was the method used in the Pangenome project that Martin just talked about. And I cannot see my pointer. 1 second, sorry. Oh, okay, let's try without the pointer. So this work was published in 2022. And if you have any questions, you can, of course, contact us, check the paper. And all right, let's go. So this is an overview of how Tritex works. You need 3 kinds of data sets. So a guide map, that can either be derived from a reference genome assembly or from a genetic map. Long reads, we used PacBio hi-fi reads, but if you can obtain a good config assembly, it's okay. Doesn't matter the type of data you're using, just matters if the config assembly is good. And also the high-C reads. So the first step is obtaining this config assembly, and then the third is to map the markers to this config assembly and the high-C reads to this config assembly. Then these three first steps in red are done using Unix scripts and also other bioinformatics software. And then to unify all this data, we create this assembly object now using R in blue, the fourth step. And now we can curate the assembly. So using different kinds of visualization, you can do the scaffolding part of your assembly. And then when the assembly is looking good, you can generate the pseudomolecules and you will have the fastest sequence for your genome. So Tritex was already used in many species, either for a single genome or for more genomes, like Martin just showed, so for the Barley Pangenome. So many cereals: barley, wheat, rye, egglops, lolion, oats, eggplant, faba bean, which has a very big genome also, pea, rapeseed, and maize. So the next results I will show you were done in maize. And the first thing we do is to check if the config assembly has any chimeric scaffolds. So here I show you an example of a chimera. So in the first plot on the left side, we have a chimera of chromosomes 3 and 6. And this is quite clear in the first and the fourth graph. The fourth graph is showing a drop in the high-C coverage, which shows that this is a chimera. Then you select the bins in which this chimera is. So for instance, here around 18 megabase, 19 megabase, and then you can break this chimera. So this will generate two different contigs. And then you can proceed with your assembly. When there are no more chimeras left, you can generate the high-C matrices that can help you visualize if the assembly, if the contigs are in the right order or if there is something weird going on. And also in Tritex, we have this collinearity plot, so the second row, that help you also visualize if there are any inversions or any contig misplaced. These collinearity plots, they show the genetic map, so the guide map against the assembly you have now. So you can spot any incongruences here. These plots show the already final version of the assembly, but later on I will give you more examples of how this might look like when something is not yet correct. So for the manual curation in Tritex, we use an Excel table. So basically, on option A, we have here an inverted config. So the last one on the upper right corner is inverted because you can see that there is a different pattern. So there is like a square, and this is very uneven. And when you check the ready assembly, you can see that this has improved. So to do this, it's very simple. One must only invert the signal of the contig you need to invert. So basically change the orientation of this contig. And then upload again this Excel table and generate new Hi-C maps to check if this was enough and if there are any other changes that need to be done. Now for letter B, in the lower part, we have a misplaced contig. So the one more in the middle, so the fourth one. And simply to change this place, you can move this row to the right position. So here, simply moving the row to the last position will generate this more even Hi-C map. And then you have the correct orientation of your chromosome. All right, then, like I said in the beginning, there are two main options you can use to assemble your genome with Tritex. One is using a reference genome as the guide map. So in this case, for maize, we used the reference assembly version 5. And the density of the markers is much higher, like we can see in the left-hand plot. But one can also use a genetic map. So in this case, we use a linkage map of this IBM population. And yes, the density is a bit lower, but it's still possible to assemble a good quality genome with this kind of data. In the table, what I would like to highlight is the second row, the number of contigs. So we have 10 plus 808 for the reference genome map-based assembly, and 10 plus 810 in the genetic map. This means that we have 10 chromosomes plus a number of unanchored contigs. And the difference between the reference-based and the genetic map-based assemblies is simply that two contigs were more difficult to place using the genetic map as a reference, let's say. So they were not included in the chromosomes, they were included in the unanchored chromosomes instead. The difference is that maybe if you have a genetic map, so a lower density of markers, you might need more cycles of inspecting your Hi-C matrix, the collinearity plots, to correctly place the contigs in your chromosome. So this is the difference. The first option here is a bit more simple, but it's also possible to use the genetic map with no problem. Even if you have less data, so we tested here downsampling the amount of Hi-C links and the number of markers, and we still get good results, even with less coverage. So for the left side, we decreased 10-fold, 100-fold, and 1,000-fold the number of Hi-C links. And for the 10-fold and 100, we can see that the orientation is still the same. The result is the same. But when we decrease too much the number of Hi-C links, then it becomes a problem. As for the number of markers, the difference was not so extreme like we see in the Hi-C links. So even if we decrease a lot, we still have a good result. Of course, like I said just now, you might need a few more cycles of inspecting these plots to correctly position the contigs. So how to use Tritex? We have also this tutorial online that is showing step-by-step all the needed things you need to do and all the software you need to install. And like I said, you can also send us questions if you have any problems using Tritex. And also another thing is that we have this map inspector Shiny app in R, and you can zoom in into the Hi-C matrices to see exactly the context in which you need to invert. And yes, so I would like to thank Martin, the supervisor, and also the other co-authors and all the group, IPK, and the funding agencies. And now I would like to show you a bit more in detail this data I mentioned. So first, I will start with the breaking the chimeras. So here is an example file of the, just 1 second, of the chimera plots. So here, this is a PDF of 111 pages. And basically, yes, you need to check one by one, but this is, after the first time you do this, it's a very simple process. You just need to basically check this, for example, is fine, no chimeras in this config. Also here, nothing wrong, but here, this is the same I showed in the presentation. We have a chimera, so chromosome 3 and 6, and here this drop in Hi-C coverage. Then basically you need to note down the page that this is happening and put this in the code. But this you can check with more details in the tutorial. Here, when we have this kind of pattern with just a little dot, this is nothing to be concerned about. Might be only a repetitive region, but this is what we are looking for. So this drops in Hi-C coverage. And then after you select all the ones you have, I already did this before, so I can tell you that these are the only 3 we have in this genome assembly. Mostly because PacBio Hi-Fi reads don't generate a lot of chimeras. The quality is very good, so yes. And then you can see here the marked positions in which this config will be broken. So these are the places in which we have to generate new contigs from this one. Good. Then after you have the chimeras broken, you can go and check your Hi-C matrices. And the Hi-C matrices are interchromosomal, so here in the left side, or intrachromosomal, checking the interactions between the chromosomes. So here, in the right side, I would like to quickly show you this little pattern here. So this kind of things is what we are looking for. So here we see a strange pattern in our Hi-C contact matrix. And we see that there is an interaction with another chromosome. And here we have chromosome 3 and 5. So then if you want to take a closer look in each chromosome, this is how you do it. So 1 page per chromosome, and then you can search for different patterns and to check if there's something inverted. So for example, here, there is this white line, and this indicates that this config is inverted, because otherwise this pattern should be a bit more even. I will show you how it looks after changing it, just a second. And then here, this is the same I was showing before. So here in chromosome 3, this area that is probably a config that needs to be moved to another chromosome. And Yes, one thing I forgot to mention is that these gray bars here show the borders of the contigs. So we have, for example, 1, 2, 3, 4, 5 contigs, and so on. So this helps you to localize yourself in the chromosome. And so this contig here needs to be moved. Also, here we have a very clear pattern showing that this last contig here needs to be inverted, because we see this kind of break in the pattern of the matrix. And also here we see these little red points here in the border of the contig. And this indicates that they need to be changed. So, all right, now we know more or less some things we need to change, but how do we do that? So basically, you can open this Excel table that has, I don't know if it's too small, I'm sorry. I hope you can see well. So the first sheet shows us the unanchored contigs. So these are all the ones that were not assigned to any chromosome. And the other sheets show each chromosome. So here for chromosome 1, we didn't have anything concerning to change, so we don't do anything. Now for chromosome 2, like I mentioned, the first contig needs to be inverted, and then we can simply come here, so the last column, and put, instead of 1, you put -1. And this will make the inversion of the contig. And here for chromosome 3, like we discussed, this contig here needs to be removed or placed in another chromosome. So we checked here in this other plot that this interaction is probably with chromosome, I said 5, but it's 6, sorry. So here's 6. Then basically you can see it's 1, 2, 3. So 1, 2, 3 from the end to the beginning. And then you can remove it from here. And then you can correct the number of bins. So here, this was 9. Now this will be 9, and this will be 10. We cut this and put in chromosome 6 page. And now I will not necessarily put in the right place, but I will just show you how to do it. You put this information here. Again, you need to correct the bin numbers. So this will be 4, and the rest will be 5 and 6 and so on. And that's it. So of course, I'm not going to do all the manual curation for this genome here, but also something that I showed you was in chromosome 5, first and last contig should be inverted. So here it's 1, becomes -1, and then the last one also -1. Then we can save this with a different name and upload this in R again, compute the new matrices, and check if our changes were enough. All right, I showed you how to spot these inversions looking only at the Hi-C matrix, but here we have the collinearity plots that can also be a good help. I'm sorry, this is the wrong one. 1 second. I don't find the correct one, but I can quickly show you later when I find it. But anyway, this is how the contact matrices, the interchromosomal matrices will look like once you remove this. Remember the contact that was here? It was removed. And now zooming in a little bit in the new IC matrix for the chromosome 3, we see a more even pattern. You might have spotted this little incongruence here in the IC matrix pattern, but because this is in the middle of a contact, so we see this because these are the borders of this contact. This is nothing to be concerned about. This is basically just a drop in the IC coverage. This is normal, especially in repetitive regions. So it's okay, nothing to be concerned about. Here in chromosome 5, we inverted. So let me show you both plots. So we can clearly see the improvement. So this was a super square and super clear inversion here also. So now we have this more smooth pattern, and now we can proceed. If there is nothing else that we need to change in this assembly, we can proceed to generate the pseudomolecule. And I think this is more or less all I wanted to show you. And of course, in the first time that you do this will be a bit confusing and many doubts will appear, but the more you do it, the easier it gets. So that's it. I can take any questions now if you have. Thank you.
Thank you, Marina. It was an excellent. Thank you for the tutorial demonstration. It was really useful, I think. So we already have some questions in the chat. So Robert Waterhouse has a question for Martin. So did the Barley Pangenome show any evidence of a reduced immune system in domesticated varieties?
Short answer, yes. There's a bit of a problem with the immune system because plant pathologists or some plant pathologists don't like the word immune system. But of course, we have resistance genes in barley and wild and domesticated barley, and they often occur in complex gene clusters. And this was one of the foci of our study. So we looked at these complex loci that were resolved with long-read sequencing. And indeed, we saw more haplotype diversity in wild barley than in domesticated barley. As with the amylase, the challenge is now how does this translate into phenotypic diversity? Because often these gene clusters are NLR genes, so nucleotides binding leucine-rich repeat genes that work in a gene-for-gene manner. So it really depends on the pathogens that challenge the plant. And often we see in our plant genetic resources that they are not adapted to the current plants and the current pathogens that are on the fields because they are just growing in the gene bank. But at least at the sequence level, we saw a reduction of diversity in domesticated barley.
Okay, thank you. Then we have a question from Tom Brown. I guess it's for both of you. Given what Martin showed regarding translocation between accessions, does it always make sense to break chimeric contigs in the Tritex pipeline?
Yes, you need to check the plots. You can also check the high C matrices in the first step to see if the high C matrix show even pattern, then there might be a translocation, and then you don't need to break the chimeric scaffold. But then you need to use all the information together to make this decision.
Yes, thank you. So I have also questions to you, maybe Martin, about the Barley Pangenome. So you have an amazing data set that compares the domesticated accessions from diverse regions, and then you have also the wild counterpart. And so this is an amazing data set that can give you important insights, not only about the domestication process, right, by comparing with the wild counterpart, but then also, and you showed that as well in the first PCA, that there is clear regional structure of the domesticated breeds. So my question is very general. Have you find interesting evidence not only of something that is common to all domesticated breeds and different? Well, you mentioned already the immune system, but other loci that may indicate that may track the origin of the domestication. And then the second part of the question is, now considering the domesticated breeds, do you see evidences of local different selective pressures, like for example, in the Iberian Peninsula, there may be different breeding strategies from the East Asia, for example, and it can give you some interesting insights about how the different regions have selected these breeds.
Yeah, maybe for the first question about domestication. Yeah, so quite a lot is known about barley domestication. So we know where it was domesticated and also some of the genes that were selected. And our current understanding of barley domestication was that barley has a mosaic origin. So it was not a single wild barley population that contributed, but many wild barley, or the origins of domesticated haplotypes come from many wild populations. And actually, we looked into this more deeply, not with the Pangenome data set, because to be honest, 76 is a bit small. So we had a data set with several hundred domesticated and wild barley's short read data, and then we made a haplotype map, and we looked where do the haplotypes come from, how differentiated are they, to estimate also the divergence ages. And yeah, we got support for this mosaic origin hypothesis, and we could also show that already 10,000 years ago, there was probably a lot of gene flow going on between wild barley. So there was a founder population that was basically in place already 8,000-10,000 years ago before barley left the Fertile Crescent. So this is, of course, a very complicated picture. It's not a single linear track, like from wild to domesticated barley, a loss of diversity. And yeah, the second question, yeah, this is yeah, we found one interesting example. In barleys, we have domesticated barleys, we have two morphological types, 2-rod and 6-rod types that differ by the architecture of the inflorescence. Basically, 6-rod types have 3 times more grains per inflorescence, and they originated only in domesticated barley. And we found that at one the locus that contains a developmental gene, there's copy number variation that we see only in 6-rod domesticated barleys. It does not exist in wild barley and not in 2-rod barleys. So our current understanding is that this copy number variation has arisen only in domesticated barleys, and it may have been selected in 6-rod types because it controls the grain size, and it has a beneficial effect only in 6-rod barleys.
Very interesting. So I have now a question maybe to Marina. So the Tritex was developed especially for, well, the focus was to do assembly of plant genomes, but are there any features about the species you're assembling? So how general it is, the application of this Tritex to other organisms, or are there any special features that make it particularly useful for plant genomes or crop genomes and not others?
Yeah, here I think the most important thing is that we are talking about mainly inbred genomes.
Yeah.
So then that's why I would not use this to assemble a animal genome, for example. And also we have I think this is the general answer. And more specifically, if you're using a genetic map, then yes, this is more specific for plants. And yeah, this is the main reason. So if it's heterozygous, maybe you should use other methods. Yeah.
Yes, makes sense. Okay. Thank you. I don't know if there are any other questions in YouTube. I don't think so. So if there aren't any other questions from the audience, I just need to thank you very much, both of you, for your wonderful presentations. It was really insightful and interesting. And have a nice day and have a nice week. And thank you very much again for your talks.
Thank you for having us.
Thank you.
Bye-bye.
Bye.